Pittsburgh University. Professor Wagner is the director of the McGowan Institute for Regenerative Medicine, as well as Distinguished Professor of Surgery, Chemical Engineering, and Bioengineering at the University of Pittsburgh. He serves as Scientific Director of the NSF Engineering Research Center on Revolutionizing Metallic Biomaterials and Chief Science Officer for the Armed Forces Institute of Regenerative Medicine. His research has generated numerous patents, 36 issued to date, and patent filings that have resulted in licensing activity, the formation of 2 companies, one of which initiated 2 clinical trials. He's the founding editor and editor-in-chief of one of the leading biomaterials journals, Acta Biomaterialia. He is past president of the American Society for Artificial Internal Organs and past chairman of the Tissue Engineering and Regenerative Medicine International Society Americas region. He is a fellow and former vice president of the American Institute for Medical and Biological Engineering and has been elected a fellow of the Biomedical Engineering Society, the International Union of Societies for Biomaterial Science and Engineering, TERMIS, and the American Heart Association. In 2006, he was elected to the Scientific American 50, the magazine's annual list recognizing leaders in science and technology from the research, business, and policy fields. He has won many awards, some of which include the Society of Biomaterials Clemson Award for Applied Research, Chancellor's Distinguished Research Award from the University of Pittsburgh, and the Senior Investigator Award from TERMIS Americas. In 2017, he was inducted as a Fellow of the National Academy of Inventors, and in 2018, he was named Inventor of the Year by Pittsburgh Intellectual Property Law Association. In 2021, Dr. Wagner received the Founders Award from the Society for Biomaterials and was elected as a Fellow in the International Academy of Medical and Biological Engineering. The floor is yours, sir.
Well, thank you for the kind introduction. So my research interest going way back to the beginning of my career has been heart disease. And when I started as a fresh PhD out of graduate school, I became interested in circulatory support, artificial hearts. And that kind of got me into this whole area of end-stage heart failure. And what struck me after a little bit of time working there is that there's this period where the heart is failing, is moving down a pathway towards less and less function. And to be blunt, as an engineer, not a lot is done about it. So there's a great deal that's done, obviously, pharmacologically to prevent the heart attack. There's a great deal that's done to revascularize immediately prior or hopefully immediately after a heart attack. But there's this window where the heart has been damaged by an infarct, is remodeling. There's not much that's done structurally to help that heart. And that's the area that my group has been, one of the areas my group has been working in for many years. And that's what I'm going to talk about today, because it looks at biomaterials and says, well, if we wanted to intervene mechanically and structurally, what materials would we use? And I think it's obvious, or was obvious back then, that the off-the-shelf materials available, a lot of FDA-approved type devices, are not ideal for the application, and new ones need to be developed. So let me see here. So this is the scenario of the failing heart. So you have the left ventricle here, mitral valve, an infarct or heart attack has occurred at the apex. And what's happening is this infarct will normally remodel and the ventricle will remodel into an enlarged heart. The mitral valve will regurgitate, it won't be able to close all the way, and the wall gets thin and cardiac output goes down. So what if we placed an elastic patch to mechanically support, just a simple mechanical support onto that wall to change the stress profile in the heart? And would that change the way it remodels? So I'll argue that yes, it can change the way it remodels. Same idea, different approach, different material, is to say, okay, you have the infarct, again, an apical infarct. We know it's going to remodel, the wall's going to get thinner, the infarct is going to spread, ventricle is going to get bigger, function will go down. What if we injected material into the wall? And it would do the same thing as a bulking agent. It would reduce the wall stress by increasing the volume of the ventricular wall. And that's an approach that's been clinically attempted now by a number of different groups and companies using all different kinds of materials. My belief is that it's critical that that material be mechanically robust. It be an elastomer, not just a low viscosity fluid. It really needs to have some mechanical support. And there's models from various groups that have shown that, in fact, mechanics is important of what you inject. So that's really what I'm going to talk about today is how do you make the patch, what do you make the patch out of, and how do you make the injectate? And looking at studies of what happens when you apply those. So this is almost 20 years old now. We started thinking about the patches, but thinking about other cardiovascular materials as well, and realized that there really weren't good options for biodegradable elastomers. There were elastomers and there were biodegradables, and polyesters were common, but they were fairly stiff. So we wanted something that was elastic and degradable. And we moved to polyurethane chemistry because it's, so flexible, pardon the pun. It's so easy to work with and interchange different components that you can really design to your heart's content, bad pun again, different types of elastomers that degrade at different rates, that have different stiffness, that have different bioactivity, and they're physical crosslinks. So they're thermoplastic elastomers, meaning you can process them with solvent quite easily. So this is one that uses a soft segment of a common polyester polycaprolactone-diol. And the ester bonds in here will degrade. We chain extend with diamine putrescine. We avoid aromatic diisocyanates or aromatic chain extenders due to potential toxicity concerns. And everything here breaks down into biocompatible byproducts. So let's get back to the heart. So this is a rat heart, looks like a human heart at this scale. The widowmaker, the left anterior descending artery, which commonly becomes blocked in humans, it's easy to reproduce that in a rat. You go in, you open the chest, and you get some suture and you tie off the artery. The lower you tie it off, the smaller the infarct. The higher you tie it off, the bigger the infarct. Too high and the animal will die. So we create reproducible apical infarcts, and then we wait 2 weeks, and it's important. You see some of these cardiac infarct models where they do something and then they inject cells like 1 hour later, or they apply something within the first, the same procedure within a few minutes. Completely irrelevant clinically. No one's going to show up and say, open my chest to be ready to do the therapy because I'm going to have a heart attack here in about 45 minutes. Patients aren't going to show up even in that golden hour. They're not going to necessarily show up. And even when they do show up, clinicians are going to be reticent to want to frankly mess with a heart that's highly inflammatory, infarcted, actively remodeling and fragile. They'd much rather prefer coming in at a point that's maybe a couple weeks down the road. So that's why we did this 2-week time point. The tough thing about 2 weeks is the heart has remodeled a fair amount and lost a fair amount of function. So it's harder to get back up to where you were, but clinically relevant. So anyhow, we come in at 2 weeks, we put on a patch, and we wait 2 months, and we look at it. So this is now, this is the infarct. You see the suture line, you see the discoloration, classic infarct. If you open that heart up, you see the wall is thinned out, just like that cartoon I showed. The ventricle is dilated. Over here, patch was applied 8 weeks ago from this picture. And you can see this wall compared to that wall is thicker. I can tell you mechanically, it's softer, and that this ventricle is more functional, as we'll see when the animal is alive. If you look at the histology, you get a very different picture. Here, again, classic, thinned out, lots of collagen, scar tissue, a few fibroblasts, a few blood vessels, not much going on. Here, you have remnants of the patch, a lot of macrophages, a lot of CD68 positive cells up here. Below, you have an interesting cell type. It's contractile smooth muscle-like, similar markers, some embryonic cardiac markers. I'm not going to get into it, but an interesting cellularization. But biologically active tissue, lots of cells, more vasculature, and mechanically softer and thicker. So it's not that this part of the heart is now suddenly going to start contracting in sync with the rest of the heart, but it's not going to be the same barrier or break to the contraction for the rest of the heart. It's going to play nicer with the rest of the heart and help that rest of the heart do what it needs to do. And it's preventing that infarct from continuing to grow and the scar to grow, which we'll see later. So echocardiography, we're interested in basically how much blood the heart is pumping. And with echo, you can do a two-dimensional slice, and basically you get diastole and systole, and you make those traces and you say, what's the difference between diastole and systole? Bigger difference, more blood being pumped, healthier heart. Smaller movements, sick heart. So what you see when you do the infarction control with a sham surgery is the end diastolic area. So in diastole, how big is the heart? It continues to enlarge, continues to get bigger. It's already, this pre is 2 weeks post-infarct, so it's already enlarged a bit. Here with a patch, you hold it steady. If you look at fractional area change, that's the difference between systole and diastole in terms of the area. And a bigger number is better there. And a healthy animal would be up in here. So at 2 weeks, we've lost a lot. Actually increase it a little bit with the patch. And if you don't put on the patch, you continue to deteriorate. And eventually these animals would die early because of the infarct. So the next level test that we did, and this is still now several years ago, but we were excited by that. And we said, well, what about functional reserve? Because it's not just when you're resting how your heart is performing it. You know, the cardiologist is going to want to know the stress response. When you go up a flight of stairs or when you're walking, are you able to keep your breath? Are you able to increase your cardiac output in response to metabolic demand? So a healthy heart, a healthy rat in this case, when you hit them with dobutamine, they will have that stress response and they will pump more blood. The fractional area change will go up. With an infarcted rat heart or human heart, when you stress, the cardiac output goes down. There's no more gas in the tank. That heart is already maximized and it loses function. And what we found interesting is with the patch approach, we actually had a functional reserve, which is a benefit on top of the benefit of having higher output. So very encouraging. And that's really speaking not to what the infarct is, but to the preservation and the health of that remaining myocardium, which is going to carry the load for the rest of that patient's or animal's life. So rats are small, better than mice, but not nearly human size. So we went to a pig model. And anyone who's worked with pig infarcts knows how delicate they are. They fibrillate, they die, they're expensive. You don't like to have many failures. So we went to a model where we only did one thoracotomy. So we started off with a balloon-based ischemia reperfusion. So we inflated a balloon. You see it in this radiograph here. And then we occluded the left circumflex artery. So this is a little higher up on the heart, left circumflex for about an hour and then reperfused. It's a smaller infarct. It's not nearly as bad as what we were doing to the rats. But same thing, at 2 weeks we patched and then we did follow-up. And you can see here, here's the pig-sized patch. We use a thermally induced phase separation to make the patch, stress strain behavior. And this is at 2 weeks now, we've opened the heart up. We're stabilizing it with the octopus device to anchor onto the apex. And then at the end of this open chest procedure, you can see the patch has been placed here. It's been sutured onto the epicardium. And you'll see in systole it'll wrinkle, and in diastole it stretches a bit. So that's that mechanical, material mechanical support that's being applied to that heart. If you look at the echocardiography, similar to the rat, we did a sham for control sham surgery. And if you put on the patch, you hold the area constant, it continues to dilate if you do nothing. And if you look at the fractional area change, you lose function without the patch and you hold function with the patch. Smaller differences because, again, it's a smaller infarct, But of note, one of the things you'll see clinically with a dilating heart is this mitral valve regurgitation, and that clearly was apparent only in the sham group. We did not see that with the patch. We prevented that in the sham group. Most animals had regurg. Okay, so still on the patch, we started asking questions. How stiff should the patch be? How long should it be there? How long should the degradation time be? What if we combine it with cells? What if we combine it with growth factors? Things, what else can we do to really optimize this therapy? So one fundamental question was degradation time. We use polyester urethane urea with polycaprolactone soft segment. It's fairly easy to play with that chemistry, as I said before, and replace the caprolactone with hexamethylene carbonate. So ester versus carbonate. Carbonate degrades slower than ester by hydrolysis. And we did some subcutaneous rat implants and showed that we could get a very nice predictable rate of degradation change as we increase the carbonate content. More carbonate, 100% carbonate, slow. Caprolactone, fast, relatively. So we had this knob that we could tune the degradation rate. So we looked at 3 different polyurethanes. The polyester, which is fast degrading relatively, polycarbonate, which was the slowest degrading, and a 50-50 molar blend of the 2 in the soft segments. And that was in between the 2 in degradation rates. And the answer that we were looking for became clear that the 2 slower degrading ones were equivalent statistically, and they were both better than the faster degrading patch. So you see here, end diastolic area, this is that rat model. The slower degrading patches do better than the faster degrading patch. And this is cardiac output here, fractional area change, same thing. So I'm not showing you, people ask, what about if it never degrades? If you put in a EPTFE patch, for instance, it prevents dilatation, but your function is, through the floor. It doesn't do anything for function, and you deteriorate actually a little bit quicker than doing nothing. Okay, so bioactivity. One regulatory-wise attractive thing to do is to work with decellularized matrices. And I live down the hallway from Steve Badalak, who many of you know. He's a pioneer in decellularized matrices, and he's been working a lot with hydrogels derived from ECMs, from extracellular matrices. And we worked with his group, and we're decellularizing hearts and then making a hydrogel from this by enzymatic degradation into hydrogel. And then we took that cardiac ECM hydrogel and we electrosprayed it while we were electrospinning polyester carbonate urethane urea. And by controlling the speed of the drum, we could even make it anisotropic and put it in the heart in different orientations. I'm not going to go into the orientation stuff. I'm just going to focus on the patch. So what we did is we made what we call an open face sandwich. Initially, we did polymer with little to no ECM to make like a sandwich bread type side. And then we cranked up the ECM, which you see in blue here, to make a lower layer that was rich in jelly, kind of like peanut butter and jelly. And if you know Murphy's Law, if you have an open face peanut butter and jelly and it drops, it lands jelly side down 100% of the time, right? That's the law. So what we wanted to do is that same thing with the jelly side down onto the heart. So we wanted ECM side rich down onto the heart, because that would allow those factors preferentially to diffuse and transport towards the heart and encourage cells to grow up from the heart. So that's what we did. I'm not going to show the data, but basically compared to a control without the ECM, reduce scarring, increase vascularity, improve cardiac function, all these things. So similar to the patch data I showed you before, but we are doing electrospinning now instead of thermally induced phase separation. So a more modern take on that, or a more recent take on that, is I had a visitor from Japan in my group, Dr. Kashiyama, and he had worked with a number of the Japanese groups that make cell sheets. And so we made an adipose derived stem cell sheet And that has actually been used clinically. They've applied that to infarcts. And we wanted to know what would happen if we took the stem cell sheet and put it below our open face sandwich. So theoretically, having the ECM on top of the cells, on top of the infarct, seemed like a really nice combination. And then the polymer giving mechanical benefit. So this is from that paper. It's in press but available through Journal of Thoracic and Cardiovascular Surgery. And you can see the three groups. So basically a stem cell patch, a stem cell layer with the PECUU patch versus each one of those individually. And one thing, I'm not going to show all the data, but one thing was the cells survive a lot better when they're underneath that ECM-laden patch. So we had more cell survival at 8 weeks out. And then if you look at the data here, basically the green, this is end diastolic volume, so lower is better here. This is patch alone or cell sheet alone, and same thing here, and systolic volume. Basically what you see is when you have the combination, you improve even further. Either one of the stem cell sheet or the ECM-based patch is good, but when you add the cells, you get even more benefit. And then in terms of function, ejection fraction, you can see it's better with the combination. So from a regulatory perspective, from a production perspective, clearly more of a hassle, but at least in the animal model, it appears to be benefit there. Okay, another fellow that came through my lab was Lindenberg from Brazil, and he is a cardiac surgeon that contacted me, very interested in this end-stage heart failure. And if you know Brazil's history in this topic area, there's been a lot of innovation that's come out of Brazil in the past decades on end-stage heart failure with cardiomyoplasty type approaches. So what Lindenberg wanted to know is, what if we do this to a really blown out heart? So what if we come in 8 weeks for a rat is a long time, 8 weeks after infarction. So that wall is really thinned out, not much cellularization there. You've lost a lot of function. What if you put the patch on at that point? Is there any benefit? Because clinically, there's a fair number of patients that are going to be in that group. So that's what he did. There you can see the control hearts. Here you can see the patch treated. Again, no panacea, but what it's doing is it's helping the rest of the muscle contract and preventing further dilatation of that infarct. And if you look at the function, control group versus patch over time, up to 8 weeks, you see you continue to lose function with the control, where you actually improve the function with the patch in this group. And we did a control group of losartan, which would be a drug that would be clinically used in this scenario to try and help with remodeling. So in that study, one of the most interesting things is we looked at the sickest of sick animals. So animals that had an ejection fraction below 35% at 8 weeks. So these are rats that are, at death's door, basically. And we asked what happened with those really sick animals. And that's where the benefit was most pronounced. So you see here with the patch, again, looking at ejection fraction over time, pre all the way up to 8 weeks after. So pre, again, is 8 weeks after infarct. And then going another 8 weeks, we actually markedly improved the ejection fraction, where you continue to lose it with the control and the losartan. And not shown here, but statistically important, is there were more deaths in the control group. So if anything, you had a bias, a positive bias in the remaining survivors for the control group. Okay, so I want to shift to stop talking about patches. I've kind of updated you. There's a number of other tweaks that we've done, but you get the general picture. I think there's a fair amount of promise with this patch type approach. But what about the injection approach? The injection approach has some attractive features. I'll show you that minimally invasive approaches are a little bit easier with the patch, and we believe it's important to come from the epicardial as opposed to the endocardial side. Others would disagree with that. I'd be happy to enter into those debates, but I think epicardial is the way to go for, at least for our materials. And you're not having to do a fixing of a patch onto the epicardium, which could be done minimally invasively, but it's a lot easier just to go and inject. So this is a concept. Again, we want to get gel, mechanically robust gel, into the cardiac wall. So what do you inject? So if you sit down with a whiteboard at the very beginning, you say, well, ideally, we'd like to inject through a very small needle. So that means low viscosity. So we want low viscosity material, but when it gets into the heart, into the cardiac wall, we want it to be elastic. So that means phase change or reaction, right? You're going to cross-link, or you're going to do something to make it change mechanical properties. But then we want it to go away. We don't want to put a permanent foreign body in there. We think that that's just going to create a fibrotic area that in the long term may not be as beneficial. So we want it to be there for some period of time. We don't know how long, but we want it to be there and then go away. So here's how we did it. We used the N-isopropyl acrylamide chemistry, which is well known in the field. And that has a propyl group that gives you this inverted thermal responsiveness. So basically, as you raise the temperature, it collapses. It sheds its water and collapses hydrophobically. So we have a backbone here that has isopropyl groups on it, and isopropyl acrylamide, and some other groups. But then we have these side branches that are hydrophobic, and they have a labile, at least one labile group associated with them. So you raise the temperature, these will collapse. So that's, design parameter number one. It goes from low viscosity to being elastomer or viscoelastic. And then the second part, you cleave naturally hydrolytically the hydrophobic branches, and that ends up effectively raising the lower critical solution temperature. So you're changing the temperature at which that phase change occurs, you're raising it, and it gets above body temperature and it becomes soluble again. And then it diffuses away. So we're degrading off side branches to get to the point where we can solubilize the backbone. And then the nice thing about the side branches is it allows this backbone to be lower molecular weight, so you can get it below the point at which it's going to pass through the kidneys to minimize toxicity concerns. So lots of ways to do this. I'm not going to go into all of them. Here's an example though. So we make this is the pink, this pink side branch is right here, and it's hanging down here. So trimethyl carbonate. So the carbonate is going to be the labile group. That's going to get cleaved. And then when that gets cleaved, these pink groups come off, and then this becomes soluble. Here's your N-isopropyl acrylamide, and this is just a hydrophilic acrylic acid that helps in the tuning of this. So this just shows, the PTMC is that pink group. So here we have it on the polymer, and here we've cleaved it off. We've gone ahead chemically and cleaved it all off. And so you see, this is what it would be like when it's injected, using different molar ratios of this to make the addition polymer. And this would be after we cleaved it off. So the point is, body temperature at 37, this one is not relevant because it does not go through the phase change until way above body temperature. So that one's out. These three are possibilities. They all will gel in the body. And now after we've cleaved this off, we want them to be on the other side. So these two, the red and blue, are candidates for being used because they do exactly what we want. They gel, and then when they degrade, they will diffuse away. So that's basically the material design, or one aspect of material design. Here's an example of one of those polymers in the syringe. You can see this is 37-degree saline. That's real time. So when you inject, it's it's doing that phase change quickly, which is what you need. You can't, 2 minutes is too long. You can't do injections and then wait and hope it gels. You need it to gel quickly. Even with quick gelling, you're still going to have some regurgitation. So a little bit of regurg is okay, but too much is not good. Here's another example of some of the chemistry. So here we have 4 MERs that we're putting together, and we're changing the ratio of the methacrylic acid and the methacrylic acid and the HEMA. So by changing that ratio and increasing the methacrylic acid, that is going to catalyze the cleavage of this polylactide right here. And that's the equivalent in this polymer, that pink strand. So the more of this, the more it should auto-catalyze and the quicker it should degrade. So what this is showing is that we can design polymers that all have a pretty similar phase change behavior. They're all plus or minus 2.5 degrees Celsius in terms of their phase change, but they have radically different degradation times. So with low methacrylic acid or 0, you can see the degradation time is way out here. And with high, it's very quick, a day or so. So we can tune by changing those molar ratios. We can do like we did with a polymer with ester and carbonate. We can change the rate of degradation. Okay, so in vivo data. Here is the same rat model, the same ligation that we'd do before, except now at 2 weeks, when we open up the chest, as you see right here, we're injecting instead of putting the patch on. So generally injection in the middle of the infarct as well as the periphery. And that's going to be true with pigs, with rats. You know, we always do that. We cover the infarct area and the border zone, right in that border zone. We don't do the whole open surface of the ventricle, which some people have done clinically and in some large animal models. Don't see a reason to do that. You don't want to mess with relatively healthy myocardium. So here's that echocardiographic data like before. In diastolic area, hydrogel injection, you pretty much hold that steady. Our control here is saline. So very similar procedure, except now we're just injecting a similar volume of saline. And then if you look at the function, you hold it steady with the gel and you lose function if you put in saline. We've done a number of studies with different gels and asked some of the questions. One of the questions that we had, we came to after some discussions with one of our cardiology collaborators, and it was this time of intervention. And he really impressed upon me the reticence for the clinician to intervene in that early period. And he said, 2 weeks is great. And I said, well, we'd like to back it up because we think we're going to get more benefit the earlier. He said, well, maybe 3 days, they might entertain that, but it'd have to be much better than 2 weeks. 2 weeks is good. And even later, would be better. So you're doing this trade-off between losing function in the patient and putting the patient at risk for an intervention. So we did a study with the injection comparing the one I kind of poked fun at earlier, immediately after MI. So you do the MI and then you immediately do the injection. So clinically irrelevant, but theoretically, maybe that would be by far the best thing to do because you haven't lost any function, right? There's no function loss right after the infarct. So you inject right at that point. Then we did 3 days after, which we thought was the closest in clinically that might work. We did 1 week and we did 2 weeks. And you can see the substrate that you're injecting into is quite different. So this is what the heart looks like that you're injecting to. And you can see at 2 weeks that you've got a pretty well-developed infarct there. At immediately post-MI, it still looks okay. You haven't had the necrosis and the inflammation set in yet. And here at 3 days, you begin to see that happening. So here's the data. It's a little bit confusing. This is time after MI. So at some of these, the injection is going to happen at a later time point than at others. But the key thing is just looking at the colors. You can see immediate is blue here, 3 days, and then control MI, so it's no intervention. So the one that jumps out is the red one. 3 days. And if you compare 3 days to immediate, you see the immediate wasn't good. The immediate was almost as bad as the MI control, equivalent to the MI control. So mechanically putting that material in there, at that time point into that substrate where so much is going on doesn't get you any benefit. But if you wait 3 days, you maximize benefit. If you wait longer, then you're losing more functionality. And we did some assessment of inflammatory markers. And indeed, at 3 days after MI, TNF alpha, for instance, is lower. IL-1 beta is lower. So it's a different substrate that you're working with when you do those injections. Even if you do it and then look at it later, you've changed that environment. Okay, so more recently with the injection, we've, just like with the patch, we've moved up to the PIG model. And this is an open chest. I'll show you a minimally invasive way that we're working on. This is the open chest. And here you can see what it looks like. So in this model, we did not do the balloon. We actually did an occlusion. We went in and we ligated a coronary artery. So it's a little bit different, a little bigger infarct than we had with the patch. But that gave you an idea of what it's like. So we have a number of syringes and go in and do those injections. And this was published a couple of years ago. Really nice study by Yang Zhu and Dr. Matsumura in my group. And we did echo, but we also did MRI. We were able to get into the magnet after hours and with these pigs and do some really clinically relevant measurements of scar size. So this you see with saline PBS, long axis and short axis. This is the heart right here. And that discolored area bordered by the 2 yellow arrows, that's the infarct. So you see it with the saline. And then you see it with the gel. And you can see, even at this scale, the lines, these areas of scar are smaller with the gel injection than with the saline. So in effect, what's happening is the gel injection is minimizing the progression of what becomes scar. And you can see that down here when we've quantified it across the animals with the gel, the scar area is smaller. And then if we look at the functional parameters, you can see cardiac index is like ejection fraction compensating for the animal's surface area. You get that clear functional benefit with the gel. The challenge with the pigs is the pigs are growing. So the pig that you infarcted 2 weeks ago, that heart has grown, that animal has grown. So even with an infarct, sometimes that cardiac output is increasing. So that's why cardiac index is important, because you compensate a bit for that. But it's one of the challenges with the pig model. Okay, so I mentioned minimally invasive. This is work that we've done with Cam Rivera at Carnegie Mellon. And he has developed this inchworm type robot. It has two pieces, a head and a tail, and each one has a suction cup. So basically it's like an inchworm. It sucks down and moves the other one, sucks that one down, moves the other one. This is what it looks like when it moves. So it's applying that suction to keep whatever member it needs for anchorage down while it moves the other one, and then it inches along. And you can see this on a butcher shop heart, how you can steer. You can have steering wires. And then not shown here is you have markers there that you can detect. You can register where you are, so you know exactly where you are on a map of that animal's heart. And in the real world, this is a beating pig heart between the pericardial sac and the epicardium. It gives you an idea of, it's not quite as easy as that little on a piece of paper. It's a slimy, slippery, beating environment. And you have to really have a robust adhesion system to get that to work. So it's a lot harder than it looks in theory. So what we needed to do is we needed to design the hydrogel to work with that system. And the problem is the hydrogel is triggered by warmth. So we have our cool hydrogel, and you saw us injecting it into the open heart. That's easy to do. But what about when you have this robotic system? You've got to take it through a tube that's going to cross into the body. What's going to happen? It's going to change phase and you're going to gum up your line and you're going to be out of business. So we designed a coaxial system with a cooling fluid that would keep the polymer cool until the point of injection. And then after the injection, the needle would withdraw back into that cooled area and any material would go back to being soluble. So a lot of work there, 2 graduate students working on that project. And part of it was redesigning the polymer. So we introduced a vinyl pyrrolidone that you see right here and showed that we could change the temperature. And basically, we could design a vinyl pyrrolidone 15%, where we could correlate the phase change behavior of that polymer. This is the temperature at which the phase change occurs with the flow rates of the cooling fluid at different gauges of the coax. So it's a really nice chemical engineering project where you've got cooling, you've got phase change, you've got length of reactor. So neat little project, but they did a great job in getting that designed. And then we went to prove the concept that if we had a map in mind of where we wanted to inject, so if we had an MRI for that patient or fluoroscopy where we knew the infarct area, we could go in with the robot and specifically inject around that infarct and in that infarct or other places that we may want to do that. So that's what this is with the live pig. We wanted to achieve a square injection pattern. This is not an infarcted animal, just a healthy animal. And we showed that with a photoacoustic dye, indeed, we could reproduce exactly what we wanted to do. So I will wrap up. I've shown you, I've told you a lot of stories about 2 different ways to achieve the same end. And that is to deal with the failing heart after the window for revascularization has closed. So you show up in an emergency room with a heart attack, one of the first priorities is to revascularize. What that means is get blood past the blockage to that myocardium so that it does not die. So if that means a stent, balloon stent, or even bypass surgery, you're trying to revascularize to preserve as much myocardium as possible. That's objective number one. After a certain period of time, there's no point because that tissue is dead, it's remodeling. And although not exactly, clinicians generally know based on the size of the infarct, what's likely to happen. And that patient's cardiac output is going to deteriorate. When it gets to end stage, there's plenty of options. I mean, they're not great options, but there's options. You know, the gold standard is you get a heart transplant. If you're older, you may not have that option. And it's not like there's a reservoir of donated hearts ready to go. So other options are mechanical circulatory support. Blood pumps are better than they've ever been, rotary blood pumps. There's over 100 patients that have been supported for over 10 years each with a HeartMate 2 rotary blood pump. So that's changing and that's improving quality of life. But, if you ask someone, do you want to live the rest of your life with an artificial heart or a heart transplant, or would you like to have your own heart function better so that you don't need that? No brainer. Okay, so both of these techniques, at the very least, we believe, buy time. We don't have long-term data. The pigs are getting too big and the rats just don't live long enough. But we do know we can intervene at very sick hearts and still get benefit, actually more % benefit than with a more recent infarct. So it's encouraging and it's not terribly invasive. I showed you the robotic system. We can do that with relatively minimal risk to the patient, we believe. And it doesn't preclude other therapies. You can come in and put in a ventricular assist device if it doesn't work. You haven't created a big mess for the surgeon to deal with later. And then from a biomaterials perspective, forget about the clinical application. It's just a neat area to work in because you're trying to design materials to do these different things in the body and respond in the way that you want them to. So when you do that, there's other application areas. So I didn't go into a lot of our work on coatings or skeletal muscle or other areas, heart valves, but a lot of these materials can be applied to other systems in the cardiovascular area and in other areas. So that's a fun aspect of the work. So lots of people have worked in this. These are the main contributors over the past 20 years in the polymer development and in the animal models, and some of the key academic collaborators and funding. And this is part of the McGowan Institute at University of Pittsburgh, McGowan Institute for Regenerative Medicine. That institute has been supportive of a lot of the work. So I hope I've left a little bit of time, and I hope there might be some questions.
Thank you for the fascinating talk, Professor Wagner. Several questions came in. What could be a regenerative medicine approach to prevent heart attack rather than treat it? Could coronary artery blockade be targeted and reversed?
Can coronary blockage be targeted and reversed? So yes. When a coronary blockage occurs, the vast majority of the time it's a thrombus. So a fibrous cap on a coronary plaque has ruptured, and your body builds up a platelet-rich plug, and that occludes the coronary artery and causes a heart attack. So there are a number of groups working on nanoparticulate and microparticulate technology that will home to that thrombus and deliver fibrinolytic, enzymes that will break up that clot, or be triggered by ultrasound to mechanically break up that clot. So there's a number of therapies to do that, or just clinician injecting fibrinolytic drug into that area would theoretically do that. The problem is that the time between that happening and the clinician being able to do that is usually pretty long. So at that point, it may make more sense for them to just come in with mechanically with a stent. It may not be completely occluded, and try and open it up. But yeah, theoretically, that would be attractive to do that.
Thank you.
I guess I'm looking at a question. Prevent heart regenerative. Yeah, I think the other aspect to that question that is maybe a deeper aspect is, could you do something to the coronary artery that is diseased to regenerate it, to remodel it? And that is an area where I have not seen as much work. You see that more with aortic aneurysms, where people are looking at stem cell approaches to try and remodel the aorta. But I think that's an area that could have some potential, and that isn't heavily trodden right now. So it's a good point.
Thank you. Could the regenerative medicine approaches be applied to heart transplantation, particularly to prevent ischemia reperfusion injuries after organ procurement and before it is being transplanted, or to address and treat heart rejection?
Yeah, important and insightful question. So right now in organ procurement, one of the big trends is for perfusion technologies. And this is particularly hitting with the lung. So the lung seems to be the one that's the avant-garde, leading the pack. So basically what they'll do is, when they harvest from the donor, they will perfuse that organ with hemoglobin-based solution or a blood-containing solution. Maybe it's above body temp, or maybe it's at body temperature, maybe it's below, maybe it has a certain mixture within it, but they're really looking at that period where the organ is outside of the body to prevent exactly that, to prevent the ischemia and to minimize the damage. And and I think the organ that'll probably be next will probably be the liver. We had some at our institute. We had a group that did a lot of work in that with liver. And I remember looking at the histology where they did the perfusion versus where they didn't. And it was remarkable, the improvement in how the transplanted organ looked. So I think once you do that, once you get the organ outside of the body and are able to maintain it for a longer period of time, first of all, you change space and time. So now you're able to deliver that organ in a healthy state to a greater radius. So that improves the number of organs that get transplanted right there. But two, when you have that organ perfused, you now opened up the opportunity for adding value to that organ. So adding value by gene therapy, by cell therapy, by other interventions or characterizations where you really can work with and characterize that tissue. So the obvious thing is just having a healthier organ to transplant and minimizing ischemia reperfusion. But when you think about it, if you're able to do that with the donated organ, there's a lot that can be done to improve overall outcomes for organ transplantation.
Thank you. What is the advantage of PTMC as compared to other polymers like PCl or PLA? What about the PTMC toxicity compared to other polymers?
So toxicity, we haven't seen any differences with it. The advantage is it's not really an advantage, it's just slower. The carbonate bond is just generally slower degrading than the ester bond that you have in the caprolactone or the polylactide. Yeah, that's pretty much it. There's some differences in terms of when the molecular weight of the soft segment, whether you induce crystallinity in the soft segment or not. So there's things that you can do with some of the soft segments where you get crystallinity, and that can lead to some shape memory behavior that may be useful. But yeah, it's nothing profound.
Okay. Another question came in. What is the best method to check degradation of polymeric materials or biomaterials?
Well, the easiest method is weight. Easiest method is you're looking at hydrolysis and you put it in a buffered saline in your incubator, and you take it out and you put it in a drying oven and you weigh it. So that gets you a fair amount of insight, and it's certainly the first pass. But beyond that, ideally you'd like to know molecular weight, because you'll lose molecular weight before you lose mass. And it can tell you what's going on before you have a sudden mass loss. This is particularly true with hydrogels. And then the other aspect is enzymes. So a lot of the polymers that I was talking about today, you can do the hydrolysis and you'll get data at 8 and 16 weeks that will suggest maybe 10% weight loss. But if you do it in elastase or lipase, you'll get much faster degradation. And if you look at a subcutaneous implant and look at that mass loss compared to what you get in vitro, the enzyme degradation is a much better approximation of what you're going to see in vivo. So the enzymes are working in concert with the hydrolysis to accelerate degradation. And if you're talking about a biomaterial, you care about what it's going to do in vivo, not what it's going to do in a landfill or what it's going to do in your lab.
See. The last question is also related. There are many biomaterials available. What do you think the ideal biomaterials characteristics theoretically should be that would solve the problem? What is the major challenge developing those?
Well, it's a good question, and I would challenge that. I would say that there's not that many biomaterials available from a regulatory perspective. So if you look at the materials that are used in FDA approved or CE mark approved devices, it's actually a fairly small subset compared to the universe of what you see when you pick up a biomaterials journal. So in biomaterials journals, we have this beautiful chemistry. I'm as guilty as the next person of publishing. I talked about some of it today. These chemistries where we're showing how we can tune things. That's important to open up new ways of designing things, but ultimately those materials, if they're going to get to the patient, have to get through the regulatory pathway. So they're going to go through a battery of testing that's going to be expensive, very expensive. And one of the first things the CEO of the startup company is going to do is they're going to look at the material and they're going to say, okay, can I replace that with alginate, or can I replace that with collagen, or can I replace that with PGA? They're going to pick one of these standard things. And the answer may be, yes, you can do that, but you'll lose 10% of your activity. And they'll immediately think, that's probably worth it. I lose 10% activity, but I save a fortune in qualification of that material. So it's a vicious circle. I'm not saying it's wrong. I'm just saying that there's this pressure to keep using the same materials because innovative materials have this high burden of safety that you have to demonstrate. So that's the that's a major challenge. It's not the intellectual, and it's not physical or chemical. The biomaterials literature is loaded with these reports, and you've got to, if you want a good exercise, I say this in the biomaterials textbook. If you want a good exercise, go back 20 years, pick up a leading biomaterials journal, or Science or Nature for that matter, that has a biomaterials article. Read the articles and read the last paragraphs and how they talk about how it's close to clinical impact, or it's going to go to a large animal. And 9 times out of 10, and maybe there's a couple follow-up papers, but it never did that. And why? Why didn't it? Sometimes you'll see it never got a patent. A lot of times it will have gotten a patent, but there's something that someone's not telling you. I tell students this: someone's not telling you what's happening. There's this mystery out in the forest that all this technology dies, and that mystery is the translational pathway and the cost of doing that. So that's why.
Thank you for the detailed answer. That's all the questions I have. Thank you for your time, Professor. Great talk.
Thank you. I appreciate the thoughtful questions.
Thank you.